part of our activity, we discuss the external parts of invertebrates, such as insects and arachnids. A specimen was examined by us while using a microscope so that we could become acquainted and familiar with it. And insect parts, such as antenna, mouth parts, eyes, the wings, and ovipositors, were observed through a microscope. And then we were given instructions to sketch, color, name, and explain the significance of these features.
The life cycle of mosquitoes is composed of 4 stages. First, egg, larva, pupa, and lastly, adult stage. They will lay their eggs on freshwater surfaces. These eggs will hatch into larvae or wriggler. The duration of hatching will be dependent on the temperature of their water, food, and its species. The skin of the larva will tighten and shed as it matures and will stop eating. Then it will transform into pupa, or it will enter the pupal stage. It will remain on the surface of the water until it became an adult mosquito. It cannot consume food, but it can breathe through the use of the 2 tubes on its back called siphon. It would take days or even weeks for them to hatch into an adult mosquito, depending on the environment's temperature. In a tropical region, it will usually take 2 to 3 days before it transforms into an adult mosquito. On the other hand, it will take 9 to 12 days in a cooler region before the pupal stage ends. So once this stage ends, the mature mosquito will crawl into the surface of the water, waiting for its exoskeleton to harden. Once it is fully grown, it will crawl out of the water in the search for a safer place to dry and spread its wings.
Today we will discuss about the list of collecting tools for arthropods used in lab exercise 2. First is the forceps, which is delicate handling tools. Next is the vials containing preservatives, containers for specimens preservation. Next is killing bottles and small storage containers for safe specimen storage. Next is small envelopes and gel cups, which is temporary storage for delicate specimens. Next is aspirators, which is the suction device for arthropod collection. Next is absorbent tissue to prevent moisture damage in killing bottles and aspirators. Next is notebook and writing equipment for recording collection data. Next is knife and scissors used for specimen and label preparation. And small brush for handling minute specimen. Next is collecting nets used to capture arthropods, killing jars or bottles, containers for a quick euthanization, and liquid killing agents, jars, containers with liquid agent for arthropods, and baits and other attractants for substances to attract arthropods for collection.
Now, let's talk about the tools you need for collecting insects. Basic tools include nets, containers like jars or vials, and perhaps a magnifying glass to examine specimens closely. One way to collect insects is through active methods. You can use a net to catch flying insects or gently shake vegetation to dislodge them into your container. For some insects, like those hiding under rocks or in leaf litter, you can carefully lift or turn over these hiding spots. Another approach is passive collecting. You can set up traps to attract or intercept insects. Some common traps include light traps that attract nocturnal insects, sticky traps to catch flying insects, and baited pitfall traps to capture ground-dwelling species. Additionally, there are baited traps that utilize a range of attractants, such as tuna, peanut butter, seeds, and dead insects, among others. These baits are strategically placed to entice insects, making them a part of the collection without any physical intervention. The choice of bait can be quite diverse and often depends on the specific insect groups being targeted. Tuna, with its pungent aroma, might draw in scavengers and carnivorous insects. Peanut butter with its sweet and savory scent is particularly effective at luring a wide range of insects, including some butterflies and beetles. By thoughtfully selecting these attractants and placing traps strategically in the field, we can capture a wide array of insects, shedding light on their presence, abundance, and diversity in a given environment, all while minimizing any direct disturbance to their natural habitats.
For the processing of the collected species, each Ziploc specimen from a baited sample location was put into a petri dish containing 95% ethanol. Then, the samples were transferred from the first petri dish into a new petri dish that has been filled with tap water. A few empty glass slides were used wherein the specimens were placed and observed a dissecting microscope. After each observation of the specimen, the necessary data were input in an Excel sheet for the ledger. Once that everything is done, the specimens were placed in a test tube, filled with ethanol and closed with a cap.
The group was given a task of collecting various insect species found within the science building vicinity and its surrounding area. To accomplish this, we have employed both active and passive sampling techniques by utilizing sweep nets, baits, and the general hand collection method. After preservation, small insects such as ants were mounted in a paper stage to keep them steady while viewing. Larger insects, on the other hand, were pinned on a styrofoam then viewed using a stereo microscope to further examine their morphology. They were viewed in lateral, which is the most commonly used view, in dorsal, ventral, and in frontal view. The use of different perspectives in viewing specimens enable us to have a comprehensive morphological analysis and proper species identification, since certain features may be more prominent or distinctive from specific views.
In this lab activity, we were first tasked to be familiarized with the morphological terms of the family Formicidae. This activity is crucial before proceeding with the actual identification of ants, as the dichotomous key pertains to the morphological terms mentioned in the table. When we're already familiar with the Formicidae's morphology, we are then tasked to point mount the specimen that we've got and identify it by using the dichotomous key. That's everything that we've done in our entomology laboratory. Thank you for watching. I hope you enjoyed it as much as we enjoyed doing it.